People, it's another day, another bright day to talk seriously about matters that concerns our health, how we can live a better life, enjoy the life of abundance. One thing I know is the more you know, the better prepared you are. So I am trying as usual to expose the laboratory perspective, the medical laboratory scientist's role, and all that will benefit us. The world over, COVID-19 has been ravaging the world. Subscribe. My mission is very clear: to create the necessary interaction between the laboratory workers, the medical laboratory scientists, and the public, to educate the public more on what they have to do, to get them more familiar with the sample collection, so that when the sample is collected, the correct samples are collected and what is necessary as regards the good result, effective result will be produced. Without good and quality sample collection, the result will be, like I said, garbage in, garbage out. But when the sample is well collected, then obviously the results should be very, very effective in the treatment of the patient. COVID-19 virus is a very, very dangerous virus. We know how it has really devastated the world. The world population, over 6 million, almost 6 million people are now reported dead. You can imagine the number of people confirmed positive, going to over 355 million. Positive. When you say they are positive, confirmed positive, they roll the lab place in confirming them as positive, so that the necessary treatment can be given, can be understood. So the sample collection, like I said, maintains the World Health Organization standard, so that there is that necessary biosafety in the lab. The lab is very safe. The laboratory scientists and all lab staff must wear the PPE, and the samples are collected from the nasopharyngeal area and from the mouth too. I'll show you how that is done as we go on. The other samples that will be collected are the blood, stool, urine, and you can see the swab stick that will be used to collect these nasopharyngeal samples. This is a diagram that really shows to us where the nasopharyngeal swab will have to reach. It must go through the nose. The head must be tilted backwards to touch the back of the nose, making sure that it's touching the pharynx, and the swab is rubbed and the sample brought out and put in the bottle back into the swab stick. The same thing goes if it is the oral. You see oropharyngeal, it means from the mouth. The mouth has to be open wide so that the swab stick will reach the area of the tonsil, and it is rubbed to collect samples from that area. This is to show the picture. I designed it myself just to explain the spike protein that is on it. This is what we test for. Usually is the receptor, even the vaccine. The vaccine is targeting this spike protein. The messenger RNA produces the spike protein, which will elicit a reaction. The body itself, the immune response, getting it ready, priming it up, so that when the COVID-19 virus should enter, penetrate the body, the virus is attacked with the antibody produced against it. One of the things we must understand is that in the lab, the laboratory practice, the laboratory tests done, you can start first of all with the molecular testing. The molecular testing takes the PCR form. When we talk of polymerase chain reaction, this is the antigen slide
the antigen test slide kit that shows positive in this area. When you look at the left slide, you can see it as being positive. T stands for positive when you have this two band, but there is also the line for the IgG apart from the IgM. When it has no line showing, it's only showing the control, then it is negative. The laboratory testing also includes, like I earlier said, the PCR, polymerase chain reaction, where you amplify the nucleic acid present in the COVID, so that you can bring us a large quantity which will show positivity when there is that color change. After the molecular testing, you can go further to do the serological test, because most time after the third week the molecular testing comes out negative, but the antibody produced can still detect the presence of the antigen in the system. The viral load, when you're talking of results, is what is really taken, because the viral load increases with the PCR test. It has to replicate it. If the virus load is high, the replication number of cycle of replication is low, so you find the result as 40 and less than 40 as very strongly positive. The negative one, I say, and we must also note from this right of that the viable virus does not necessarily mean what the PCR is checking. The PCR is going after the virus that are there, whether dead or alive. The serological test is very useful in the late period of the illness. You from the serological test, ELISA-based IgG antibody tests have greater than 95% specificity for diagnosis of COVID-19. Testing of paired serum sample with the initial PCR and the second week later can further increase diagnostic accuracy. So you can understand the situation when you find the report not agreeing with the clinical presentation. You need to do more pairing and also additional tests, like I mentioned earlier. This is a summary of the whole process. The molecular test is up here, you can see, and the color. Then the antibody test, the serology, is down here where the slides are used. The summary of the procedure. We can also now understand that whatever you do, so long as it is within the standard approved by WHO, you report to the international health regulations. I mean, we have the NCDC in Nigeria. Every state send the result there. That's why the NCDC will always announce the present realities of COVID-19. And the WHO is also informed, so that you have a general view of what the present situation of testing is. The present situation of COVID-19 in Nigeria. Lagos, Nigeria remains the epicenter of the virus. All passengers from Nigeria traveling outside Nigeria are required to provide evidence of full COVID-19 vaccination or a negative COVID-19 PCR test done no longer than 48 hours before departure. This started from December 5th. Lagos State presently has resisted the fourth wave of COVID-19, and the report has it that England have stopped compulsory wearing of face masks. WHO approved treatment. We are now talking of treatment and control. My coverage does not concern the treatment, because that is beyond my scope. Anyway, anti-COVID vaccine is very useful to prevent and also as treatment. For prevention, we know the COVID-19 protocol: wear face masks, keep safe distance, and what else. The coronavirus is very heat labor. It cannot withstand the exposure to UV light or the sunlight for long. The disinfectant of surfaces, probably your working bench or even the handle, or if you want to really maintain that high level of safetyness, that is, you want to be very safety conscious, you need to disinfect with hypochlorite or the 70% ethanol. The present situation in Nigeria continues. For those arriving in Nigeria, a COVID-19 PCR test will be conducted by a designated private lab within 24 hours. If traveler test result is positive for COVID-19, the traveler will be transferred to an isolation facilities and managed according to national treatment guidelines. If test result is negative, passenger will continue with mandatory isolation at the quarantine facility until a satisfactory 7-day COVID-19 PCR test is done. So if you like what I am presenting, if you like everything I am doing, you need to encourage by subscribe. Just subscribe, press the subscribe label, click the like button, and also the notification. Thank you.